in our work, we build this like DRG genetic toolkit, which reveals the morphological and functional diversity of somatosensory neuron subtypes. So somatosensation is important for us to sense the texture, to feel the social touch, and to feel the noxious forces, and to feel the temperatures. Those root ganglion neurons are the first order neuron that receives somatosensory information. They have two axons. The central branch innervates the spinal cord and the brainstem, and the peripheral branch innervate the skin, different skin regions and the internal organs. 其实这些本来是应该有一些那个就是 animation 的，然后因为我看一下我播放的话，如果我播放的话，我就看不到，还是
哎，这个是必须全屏才能播放吗？不能
对对对，必
直接
须全屏才能播放。
哦，
但是我全屏的话，我就看不到，
看不到我们的
这
脸，
个，对对，
没关系，其实
看不到
可以播一
Discord。
下。
Okay. So early studies recorded the physiological properties of DRG neurons. So some neurons are very sensitive to light touch. So they have low threshold, and they are sensitive to mechanical stimuli. And these type of neurons are called low threshold mechanoreceptors, or LTMRs for short. And early studies also found there are neurons that have higher threshold. They are less sensitive to mechanical stimuli, and it requires like higher force to activate them. So they are called the high threshold mechanoreceptors. And there are also neurons that are sensitive to thermal stimuli, like cold or heat. And there are also neurons sensitive to both mechanical, thermal, and chemical stimuli, and they are called polymodal neurons.
我想问，就是这四种
Yeah.
neuron 都是在这个图画中间这些背根神经节这一个团里头啊，
都在这一团里的。
但是它就只生出这两个 X 嘛，就一个绿色，一个蓝色。
对，这个就是只是一个简图，
哦，
它并不是一个真正的这个因为虽然只生出一个绿色一个蓝色，但是 OK，
Simon 这边来了。
但是这四种都是有的，对。So the classification of these four types of neurons are based on the physiological properties. And in the last decades or so, the molecular identities of the LTMRs, or low threshold mechanoreceptor, have been like pretty well studied. But the molecular identity of the other subtypes are not really clear yet. And recently, a former postdoc in our lab, Dr. Nikhil Sharma, from the sequencing RNA-seq of the DRG sensory neurons, we found there are more than 16 subtypes of DRG sensory neurons. And some of the subtypes were known previously, such as most of the LTMR subtypes. And previously, we also have the mouse lines to label these neuronal subtypes for the LTMRs and for a few of the potential HTMR or nociceptor subtypes. But most of the DRG subtypes here do not have like genetic labeling for us to study them, especially for the CGRP subtypes. Previously, people think CGRP is a marker for the nociceptor or the pain sensing neurons. But now in our study, we found there are more than 7 subtypes of 7 CGRP subtypes. And most of the CGRP subtypes were unknown before in their physiological properties and functions. So we generated mouse lines to label the CGRP subtypes, and using the genetic tools newly generated, shown in red here. And there are also neurons that were known before, but do not have good genetic tool to label them, such as these two subtypes.
学长，我看不见你的鼠标。现在看到了。好的。刚刚就是我看不见吗？还是？现在可以看到了。
OK， 那我就看一下，就是这个还不是那个播放的状态，我看一下怎么在播放的状态之中可以把这个鼠标显示出来。
好的。
这个可以看到吗？
看不到。
看不到。
这是播放状态吗？因为刚刚那个好像不是播放。
OK。
现在可以看到。
现在看不到了。
现在看不到。
现在看不到。
那好像我们看到的也不是播放状态，因为我们看得到 PowerPoint 上的那个拖把。
对，好像不是在播放状态。
I see。OK。有点奇怪。就是说，这个 PowerPoint 怎么播放？我能不能就是先把我的这个 streaming 先停一下，然后再重新？
可以。
好的。
这个是不是可以看？
是的，现在是在播放状态。
OK， 就是可以看到我的鼠标了，是吧？
是的。学长怎么做到？太牛了！我之前都是得导成 PDF 格式的，不加动画。
就是我是 share screen， 而不是说 share 某个特定的 window， 但是我还是看不到大家的脸。
就看不到我们的脸，不好意思。Discord 可能不大适合用 PPT， 它比较适合大家一起看屏幕。不能听见我们声音应该就行。
对，听得见你们的声音，所以说你们有问题随时就问就好。
好的。
So listed here are the new genetic tools to label the new subtypes of DRG neurons. We crossed these lines, these driver lines, with free or flip-dependent reporter lines, or we use AAV injection to label the targeted subtypes. And we validated the labeling efficiency and specificity using RNScope shown here. And based on the quantification of the RNScope, these mouse lines passed the criteria to be specific and efficient lines. These are the lines that 你说。
不好意思，我刚刚可能 miss 掉这个 reporter 是根据那个 single cell 里头找到的 marker 做的吗？
对，就是这个 driver line，是这个 driver 的这个 molecule 是根据 single cell 里面的 marker 找到的，比如说做了这个 CRE 或者 CRE-ER line。就是因为比如说 SSTR2 CRE-ER 就可以标记 SSTR2 population， 是因为就是 SSTR2 只在这个 population 里面表达。然后就是要做这个 immunofluorescence staining 的话，然后把这个 CRE-ER 和 A14 cross 起来，或者说表达一个 CRE-dependent AAV virus。
天呐，这是不是要做很久？这么多？
哦，这个 mouse line 是我们就是找整音量做的，并不是我们自己做的。
哦，
然后，嗯，
然后那个验证是
嗯，
在是体内那个 DRG 的 imaging 吗？
这个就是那个dissect 出来之后，
哦
就是
，dissect。
fixation 之后，然后就是做 cryo section。
哦，我好好奇啊，这个 DRG 是很大吗？还是这居然可以直接剖出来？
啊，对对对，可以是说出来。毕竟我们做的是 adult，然后
哦，就是会，
第
就是有一
二句
个，就是相当于他在脊椎旁边有一个很明显的结，是吗？就这样。
哦，它是有那个28对，还是多少对？20对，就是对它是有很多对的。
哦，很多哦，对，每一节都有一对。哦，
对对对对对，
好的好
嗯，
的。哎，学长，你能看到我这边？如果我举手的话，你能看到吗？就发个表情，这样子，这样会不会
看不到，
看不到？好，那
我看不到。
我就直接说
嗯，
话吧，大家就直接说话问吧。嗯，
行行
好的。
，OK，
学长，我有个问题，
嗯，
就是您在这个 single cell 里面找的这个单一的 subtype 的一个单一的 marker，它好找吗？因为我们 lab 也在做一个 subtype specification 的 project，但是它的问题就是你能找到能在这个 subtype 里面能特意表达的，但是它不只在其中一个 subtype 里面表达，它可能会在其他的地方也有表达。我就想问一下，您是怎么筛出就是最好的这个 subtype marker 的？
哦，对，其实这个就是 hit and miss。有的 marker 虽然看上去在某一个 subtype 里面表达，但是其实做成 mouse line 之后，它 labeling 并不是 specific，就是有可能是因为在其他的 cell type 里面它是有比较低的表达，但是如果用 cree 或者 flip label 的话，这就会有 unspecific labeling，这是一方面。另一方面可能就是在 DRG 的话，它这个 genetic program 还是就是比较保守的，所以说，它这个不同的 cell type 之间的这个区别还是比较大的。另
那
外，你说，
就是接下来一步的话，不知道您是不是后面也做一些功能性的验证？就是因为这个single cell，然后通过 transcriptomic 的方法找出来的这个 cluster，它还是相当于是在算法上更加聚类的这么一群细胞，但
对对
是
对，
它的功能上是不是不太好分析其中一个 subtype 的功能？
对，所以说这个就是之后就是下几页 PPT 要讲的。
OK，
对，这个 transcriptional 方面的difference 是一方面，但是如果要 define cell type 的话，肯定是需要其他的 morphology 或者说 function 方面的 data，才可以说这真的是一个 cell type，而不是说只是一个 transcriptome 上的一个 cluster。
OK，
另外就是我们做的这个 Cree 或者 Cree line，就是因为就是 Cree line 是 inducible 的line，就是给 tamoxifen，然后之后在特定的某一个时间给 tamoxifen，它就可以开始表达，就是因为就是有些 marker 它是在 embryonic 期间表达的并不是 specific，只有在 adult 之后，它表达才开始这个restrict 到某一个 cluster 之中。所以说我们就做了 Cree line，然后在 adult 的时候给 tamoxifen。So next we want to ask what are the morphological features of the distinct DRG subtypes, and how is the mechanical and thermal information encoded by the distinct DRG subtypes? This project is a close collaboration with Dr. Nikhil Sharma, a former postdoc in our lab and a current assistant professor in Columbia University. We want to test the hypothesis that the transcriptionally defined DRG neuron subtypes have distinct morphological and physiological properties and functions. In this study, we looked at the hairy skin, the skin that covers the most part of the mouse body. And there are distinct DRG subtypes that innervates the hair follicle and the epidermis. There are Merkel cells associated, there are nerve fibers that are associated with Merkel cells. And there are also nerve fibers from the lanceolate endings, and also nerve fibers from the circumferential endings wrapping around the hair follicles. And the subtypes that form these types of endings are previously studied and known before. And in our study, we found most of our new subtypes from the free nerve endings. To get a better idea of the morphological features of the skin terminals of DRG neurons, we performed sparse labeling to study the morphological feature of single neuron morphology. So we performed a whole mount alkaline phosphatase staining, which will stain the nerve fiber in purple in a transparent or white background. So very easy to visualize under a bright field microscope. And we can reconstruct the nerve terminals formed by single neurons. So all the terms or all the gene names here are not important for you to understand this talk. So based on the sparse labeling, we found this subtype are associated with Merkel cells. They only innervate 1 or 2 hair follicles, and they have very small receptor field. And these 3 subtypes, they form lanceolate endings. They wrap around hair follicles. So these images are really zoomed out, but the morphology of these subtypes have been pretty well defined. And previous study also showed there is field LTMR, and one DRG neuron can innervate up to dozens and even 100 hair follicles, forming the circumferential endings wrapping around hair follicles. Surprisingly, one of our new population labeled by this marker, they form even more expansive innervation areas. One DRG neuron can innervate up to 300 hair follicles and covers a very large area. So shown here are the number of hair follicles innervated by a single neuron of different DRG of different hair follicles with the endings. And previously, people think free nerve ending is one type of ending, but we actually found there is great morphological diversity of free nerve endings. So these are the examples of free nerve endings formed by different DRD subtypes. So each subtype is labeled by the distinct genetic driver and reconstructed after sparse labeling. So SMR2 have this very sparse branching pattern, and one neuron can innervate up to 30 square millimeter area. And for example, this subtype has very dense branching pattern, and the innervation area is much smaller. If we quantify the branching density of these subtypes, we can see they have very distinct branching patterns. There can be can be 3 magnitudes of difference. So SMR2 has very low branching pattern, while MRGD has very high branching patterns. If we compare the innervation area or area of arborization, we can see there is also a wide distribution of the arborization areas, and each subtype has a distinct size of nerve terminals. So as this subtype has very small area of innervation, and this subtype has very large innervation areas. So there is great morphological diversity of DRD subtypes. And here we look at the peripheral terminals. for DRG neurons in the skin. We also look at, and there is one neuron subtype that we didn't see any labeling in the skin. When we count the number of cells labeled across the axial level for each DRG, we found there is a very interesting pattern. There is very few cells labeled in the lumbar or thoracic level where the limbs are innervated, but there are more innervation in the thoracic, lower thoracic, and sacral part. So we speculate they might innervate the internal organs. And indeed, although we couldn't find any labeling in the hairy skin, we found very dense labeling in internal organs such as bladder. So this population specifically innervate the internal organs.
学长，我有一个
Yeah,
技术性方面的问题，就是它这个 AP labeling，它的位置那么大的话，要怎么照啊？就是要把小鼠整个毛，就是整个皮肤照一遍吗？这个
就是那个 perfuse 之后，然后把这个 skin dissect 出来，就是把毛去掉之后，就可以做很大的 area 的，就是 imaging，staining 和 imaging。
啊，是把那个神经末梢整个的取出来吗？
对，就是把 skin 整个都取出来，然后把 skin clear 之后，它那个neural fiber 的那个 signal 就可以显出来了。
天啊，
就是因为就是 AP staining 就是可以把 neural fiber 染成黑色。
那就是他如果不把那些皮毛去掉，是不是看不见？就只有做完全部的处理之后，才能看到黑色的。
对。
天啊，那找这个 bladder 是不是很难找啊？内脏全翻一遍。
bladder 是吗？bladder 还是很容易的，就是我们就是 bladder 毕竟很薄，比较透明。
是，但是可能就不是说在找内脏的吗？那不知道是哪个内脏，是不是都要做一遍？
对，我们看了好几个内脏，就是基本上都有这个 innervation。
天，
但是就是 bladder 它这个 staining 比较漂亮，所以说我们就放了 bladder 这个 example。
还有一个问题就是它那个覆盖的那个毛孔的数量是直接算那个区域的毛孔数量，还是要看这个神经末梢有没有跟那个毛孔接触呀？
对，good question. So let me see if I can. 就是可以看到这个放大的，比如说，它这个每一个圈儿，它其实就是一个毛孔。
就是只有这种圈圈的末梢才是跟毛孔接触的，那种 free 的那种末梢就不是跟毛孔接触的。
对。
就是基本上数这个圈圈。
对。
天，太酷了！还有这个面积的算法的话，需不需要就是算一下这个小鼠的总面积之类的？就是如果它大小不一样的话，这个面积会不会差别也挺大的？
对，good question。所以说我们就是在 quantify 的时候，都是要在一个特定的 age，quantify。
就主要是按时间来。好的，哇，这个好酷啊！这个，
这样我有个问题，像那个 free end 的话，它的另一端接的是什么？还是说它直接就跟旁边的组织接触
？That is a really good question。因为现在就是之前大家也觉得是神经末梢的话，就直接跟这个直接的组织接触，比如说 keratinocytes 接触，甚至有的 paper 还说 keratinocytes 可以跟这个free nerve ending 有 interaction。最近还有一些 paper 讲，就是free nerve ending 它的 terminal 其实也有一些 Schwann cell，但是不知道那些 Schwann cell 到底在做什么。就是有些 group 就觉得，他们看到这个激活这些 Schwann cell 之后，这些 free nerve ending 也会被激活，所以说也猜这个 Schwann cell 和 free nerve ending 之间也有一些 interaction。所以说这还是一个很有意思的问题。
OK。
是不是这种 free 的末梢 是跟痛觉有关系，然后那种圆圈圈的那种是不是就跟压觉啊什么的有关系？
对对，之后会讲到，其实也不尽也不全是对，嗯，其中还就是跟它的那个 piezo2 expression，还有跟它的这些其他的 ion channel expression 有关系。
好的，
还有比如说这个 Friedner winding，它如果这个branching pattern 比较 dense 的话，它就更 sensitive；它如果越 sparse 的话，它就 less sensitive。有很多因素影响它的这个 physiological property。So we next look at the spinal cord terminals of the genetically labeled DRG subtypes. So we did a cross section of the spinal cord dorsal horn where the DRG neuron terminate. We stained for the reporter for fluorescence protein and also stained for the markers of spinal cord or different spinal cord lamina like CGRP and IB4. CGRP labels superficial lamina of the spinal cord, and IB4 labels the lamina 2 of the spinal cord. So TRPM8 subtype only projects to the very superficial part of the spinal cord, even more superficial than the CGRP layer. And these subtypes, they project to deeper part of the spinal cord around lamina 2. I think the SSTR2, BMPR1B, and SMR2, these three subtype projects to similar lamina with CGRP, while this subtype projects to a layer like closer to the IB4 band. And this MRG's family neurons labeled by these markers projects to lamina 2 overlapping with IB4 band, and CLTMR projects to the lamina deeper than lamina 2, and there are two A fiber LTMR subtype projects to even deeper of the spinal cord dorsal horn. And we can see the summarize here. So the spinal cord terminals of the genetically labeled DRG subtypes also projects to the different layer of the spinal cord. And next, we want to ask what are the physiological properties of the distinct DRG subtypes? So we performed in vivo DRG calcium imaging. Because the DRG is located next to the spinal cord, we have to anesthetize the animal, then remove the bones and muscles on the top of the DRG to expose the DRG, so we can image the calcium responses when we stimulate the leg of the mouse. Here we image the L4 DRG, and then they innervate the thigh of the mouse. So we deliver mechanical or thermal stimulation on the thigh and image the calcium response. So in this example video, we express GCAM in a population of neurons that is sensitive to the gentle stroke of hairy skin. And in this video, I will stroke different parts of the hairy skin of the mouse. So you can see different group of neurons are activated by brushing. So using similar approaches, we can identify the receptor field of the DRG neuron and apply precise mechanical stimulation on the center of the receptor field. Shown here are the 1 millinewton to 75 millinewton indentation. 1 millinewton is very light indentation, and 75 is a relatively intense mechanical stimulation. And we also apply temperature stimulation to the same area of skin using a custom-built Portier device. So, for example, this subtype are very sensitive to the step indentations. here the three traces are the three repeats of the same neuron. We can see the neuron responded early to the step indentations. So the threshold, so it has a low threshold. And if we summarize the number of neurons with different threshold that is labeled by this population, we can see most of the neurons responded to 1 to 5 millinewton, which is very low. So it is a typical low threshold mechanoreceptor population. And this subtype, they show weak activation in the relatively medium range and robust activation by the high forces. If we look at their threshold, most of the neurons are in the medium range, intermediate range of the indentation space. And this subtype only responded to the very high forces, and they have very high threshold of they have very high mechanical threshold. And in this example, they don't respond to any mechanical stimulation, but they respond to other stimuli that I will show later. So these are the 4 examples that I show, and we actually did similar experiments for all the subtypes that we have genetic access to. And the summary here are the distribution of the neurons with certain threshold. So basically, we normalize the number of neurons with each threshold and then plot it in a heat map. The brighter the color, the higher percentage of neuron with certain threshold. So we can see certain low threshold mechanoreceptor subtypes are indeed have low indentation threshold. And there are also neurons with intermediate threshold, and there are also neurons only responding to very high forces, and neurons don't respond to any mechanical stimulation. So the indentation force space is tiled as a mechanical threshold of DRG neuron subtypes. Previously, people think there are low threshold and high threshold, and the low threshold is for sensing touch, and high threshold is for sensing pain. And in this case, I actually found the distribution is continuous, not bimodal. So it suggests the encoding force might not be a labeled line, but by population coding strategy. So because we also delivered mechanical and thermal stimuli to the same piece of skin, and here there's one-fry stimuli that act as extra mechanical stimuli, and here are the cooling stimuli we delivered, and here are the heat stimuli we delivered, and each block here is a different DRD subtype, and the heat map summarizes their responses using calcium imaging. So it's a lot to look at, but if we can first look at the temperature threshold, thermal threshold. Certain subtypes don't have any response to the temperature stimuli, and this subtype only responded to the very high temperature, very high heat. And certain neurons responded to the warmth of temperature from 35 to 40 Celsius. And the trypamide neuron is very sensitive to any cooling. Just a few degrees of cooling can robustly activate trypamide neurons. And the CLTMR responding to lower temperature, and there are also neurons responding to cold. So the thermal stimulation space is also tiled at the thermal threshold of the DRG neuron subtypes. What I showed, so I just showed the DRG neurons response to the absolute force or temperatures, and we also found they encode relatively changes of force or temperatures. So when we gave the step indentations, we saw like some neurons showed the slow adapting responses like this neuron. And some neurons show the rapid adapting responses. So they show the transient response at the onset and offset of the step indentations. And most of the neurons with high threshold only show the slow adapting responses. For the temperature stimuli, we found this subtype only showed the transient response and the temperature decrease. In contrast, a subset of tryptamine neuron showed the sustained response during cooling. And interestingly, another subset of tryptamine neuron showed inhibition by the worms, suggesting they might have spontaneous activity in baseline, and worms will inhibit the spontaneous activity. So this population, this subset, and also encode the worms perception. And we also found some neurons responded to the relative increase of temperature. For example, this neuron responded to heat and also responded when the temperature goes from 5 Celsius to baseline or from 10 Celsius to baseline. So the DRG neuron subtypes encode both absolute force or temperatures and the relative changes of force or temperatures. Because we applied both mechanical and thermal stimulation, we can
学长，有个问题，能回答上一张 slide，就最后的这一个 MIG PRB4 Plus，这个 neurons 它似乎在很高的温度下才会有这个比较强的 response， 但现实生活中会有这么高的 stimuli 吗？
对，洗澡的水的话是这个大概是40到42度，然后如果到42度以上的话，就是属于比较烫的。45度，其实很多人都说我们喝的水是45度的水，50度的话就是水喝进去就有点烫嘴了，55度就是很烫嘴了。
就似乎我们 expect 是在一个就是 physiological relevant 的一个温度区间，我们还保有的这些 neurons 在进化上会有更高的一个反应的阈值，但没想到还有高温的时候也会有一些 neurons 特别的激活。
对，就是比如说你碰到一块特别烫的铁板，那就是你肯定会就是马上就把手缩回来，就是因为就是肯定有些 neuron 就专门负责感受这个特别高的温度，所以说一触到这个特别高的温度，这个 neuron 马上就要被激活了，然后你的手就能马上缩回来。
OK， 还有一个跟这个不一定直接相关的问题是，我记得我小时候看那个电视节目，他做了一个测试，说在人眼看不到的情况下，你去摸一个就是0度左右的物体，跟你摸一个热的物体，其实人产生的那个收缩反应是一样的，而且他会判断不出来它到底是冰的还是热的。
哦，有意思。对，这个确实很有意思，就是因为就是在这个 imaging 过程中，确实有一些 neuron 它只对这个非常高的温度和非常低的温度有反应，而且同时是既对这个高温也对低温有反应，所以说其实可能是这一群 neuron 就是说既被这个 cold 也被 heat 激活。
挺有意思，我就突然想到我小时候有这么一个
对，这个
不解之
谜。就是我自己就是这个 stimuli 我自己摸的时候，其实感觉也是差不多的，就是5度的时候和50度的时候，其实感觉都是比较疼，但是具体是热还是凉就没有那么明显了。
好的。
所以刚刚是那些神经元又感受机械刺激，又感受冷热刺激，是吗？
对，这是不同的神经元，所以说下一张 slides 就是说，就是哪一群神经元只感受 mechanical， 哪些神经元只感受这个只感受 thermal stimuli。
然后我还可能有一点 miss 了，就是之前是 用 life imaging 的方式，然后来确定是哪些神经元激活了，哪些神经元没激活。
对，
哦，
对，就是用 calcium imaging 的方式，这些 data 都是 calcium imaging 的数据，这 trace 就是 calcium 的 signal 的这个 intensity。
就是就是用这个神经元的 marker 和这个 calcium 的这个 marker 就是共激活这种来看是吗？
就是对，就是 calcium 的 marker 是通过 Cre 或者flip dependent 的这个 virus 表达的。
啊，就只
就是说
有要
如果
看那种神经元才能有感。好的，
对，
明白了。
So next we want to test whether the same neuron responded to mechanical or thermal stimuli. So in this population, they only responded to mechanical stimuli, so they are only in this axis. And the same is true for these three subtypes. Although this subtype also showed weak activation to cold, the amplitude is much smaller than their response to mechanical stimuli. So they are still mostly located next to the mechanical axis. And these 5 subtypes show the response to both mechanical and heat, as most of the neurons are located between these two axes. And this subtype only responded to cold stimuli, and this subtype only respond to heat stimuli. So in summary, we found there is a high transcriptional heterogeneity of DRG neurons. And from the transcriptional heterogeneity, we made mouse genetic tools to label the different DRG subtypes. Using sparse labeling, we found there is great morphological diversity, especially for the free nerve endings. And the mechanical force threshold and the thermal threshold are tiled by the different DRG neuron subtypes. So, so this work reveals the morphological and function diversity of smart sensory neuron subtypes. Okay, 这就是that's it 。
太酷了。
学长，我有一个好奇，就是接下来的这个方向，你们 lab 打算就是去 move on 其中一个 subtype，然后研究它们的生物学的功能吗？还是？
对，就是 calcium imaging，可以 indicate 它的这个 physiological property，然后可以 indicate 它的 function，比如说这个 neuron 如果只是对这个 high force，或者说对 noxious force 有反应的话，那大概率是一个就是对跟 pain 有关系的 neuron。但是我们就是之后就想通过 behavior 来验证这一点，比如说通过 optogenetic activate 这种 neuron，然后看小鼠怎么反应。就是我们可以就是跟 behavior 联系起来研究它的功能。另外一个就是看这个不同的 neuron 在不同的 organ 里面的 innervation，比如说是不是在innervate 某一些特定的 structure，然后通过这些特定的 structure 来那个推断它的功能。对，我还有一个研究，就是其中的2个 population 是 innervate genital 的一些 sensory structure，就是 innervate sex organ 的 structure，然后就是发现他们对sexual behavior 是有一些作用。
这些神经元能就是可以怎么说，就是能看它是不是保守的嘛，就是在不同物种里都能找到的嘛。
Good question。其实已经有几个实验室了，然后在看这个了。就是他们测了人的 DRG 的 single cell RC，测了猴子的，还有大鼠的，就是其实是有不少是 conserved 的 subtype。他们其中的 marker 虽然是有一些已经不一样了，但是这个 cell type 的整体还是比较 conserved。尤其是在人里面，这 CGRP 也有很多 CGRP subtype。
我还有一个很小问题，就是这个 single cell 的话，这个神经节的是做细胞核吗？还是
cell body 就是DRD neuron 就比较容易存活，
容易存活？
所以说对，就是说把那个肝杆 dissect 出来之后，用酶消化一下就可以拿到这个 single，就可以就是 dissociate 出这个 single neuron。
很专业哦。学长，之后你还会继续做这方面背根神经节吗？这方面
就是 postdoc 的时候嘛。
对啊，因为骆立群实验室做这方面吗？他不是用
不做，
果
我应该
蝇吗？
对，之后 postdoc 的时候应该不做这个。对，但是我想做一下那个小鼠的发育，然后就是 neural circuit 的发育，看这个 neural circuit 的 activity 什么时候开始出现。
activity。
对，
对，我觉得眼睛可能是最早出现的神经器官的话，因为从 morphology 它是非常快就有的结构。
眼睛，
联系上了吗？这不是？
就是小鼠就很有意思，它刚出生就是眼睛是闭着的，等14天的时候眼睛才会睁开。
但是它是最早有的。好的，那我们今天就到这里，很感谢学长给我
OK，
们
感
讲。
谢邀请。
是的，甚至还有一篇还没发呢，就跟我们分享。好的，
感谢，
谢谢，拜拜。
行，拜拜。